Hi, I'm Dr. Radha Krishna. This topic is about immunological and molecular techniques in infectious diseases. There were two questions asked in these immunological techniques for infectious diseases. The first is the in-depth broad spectrum techniques in immunological and molecular employed in the detection of infectious diseases. The second question was factors that influence immunological testing results with regards to the current practices and the future trends. So the broad spectrum in the detection of infectious diseases using immunological and molecular techniques. These immunological techniques has been used to detect both bacteria and virus infections. ELISA stands for enzyme-linked immunosorbent assay or enzyme immunoassay or EIA. This method uses plates to detect and measure hormones, peptides, proteins, and antibodies. ELISA is a widely used laboratory diagnostic method that identifies and quantifies specific antibodies, antigens, proteins, and hormones in a sample of physiological fluids. ELISA body fluids include blood, plasma, urine, saliva, cerebral spinal fluid. This methodology is used to detect infections like Lyme's disease, brucellosis, syphilis, blood-borne viruses like HIV. The time taken for the results can vary from disease to disease. It can vary from 24 hours to a few weeks. The accuracy is around 94%, 99%. This is the ELISA test. Diagram shows here that on the left side you have the control and the right side you have the samples of body fluid and the test result. The factors affecting ELISA test range from quality of the reagents, contamination of the samples, time duration, temperature of incubation processes, and improper handling of reagents and storage. So if you look at the left side, you have a current buffer system without antigen, non-coated wells. So if you look at the left side most, you can see there's blank, negative control, positive control, sample 1, sample 2, sample 3. And if you have antigen non-coated and antigen coated, the results will clearly shown on the right side. If you look at the current buffer system with antigen and non-coated, we can accurately see the specific results coming out on the right side. The next test is the Western blotting method. Western blotting is a scientific method of employing to identify, quantify the particular proteins in a sample of blood or tissues. The Western blotting involves equal protein loading, molecular weight separation, electrophoretic transfer to a suitable membrane, and antibody probing. This sample preparation for electrophoresis is essentially the downstream analysis. HIV diagnosis is usually confirmed by a wet Western blot. The test is used as a little blood sample to detect HIV antibodies, but not the viruses. So the Western blotting method, it is used to detect specific proteins from a sample of blood or tissues. It's commonly used to validate HIV diagnosis and through the identification of the virus, not the body, not the viruses, antibodies only. So if you look at the diagram, the total protein extractions is put into a tube and then gel electrophoresis, then separates out the protein, then plotting is done, the proteins are detected and then visualized. Immunofluorescence is the next test. This test is useful in detecting connective tissue disorder diseases such as lupus erythematosus, systemic discoid subacute cutaneous forms. The neonatal lupus erythematosus, systemic sclerosis. There's a mixed connective tissue diseases like dermatomyositis. But in the infectious diseases, the diagnosis of viral infections such as herpes simplex viruses or HSV or respiratory syncytial viruses RSV, influenza A, influenza B viruses, polio viruses, Coxsackie viruses, eco viruses, hepatitis B viruses, hepatitis C viruses, coronaviruses. So here it utilizes a specific paired antibody antigen to identify the deposition of abnormal molecules and proteins in the tissues or cell culture samples. This procedure can take up to five hours. Two types of immunofluorescence testing is used. One is the direct immunofluorescence techniques, where the fluorescent dye is directly attached to the main antibodies. The targeted antigen is bound by the antibodies, facilitating visualization. Indirect immunofluorescence, this process involves two steps. First, the unlabeled primary antibodies bind to the targeted antigen, so antibody antigen, and then the fluorescence is tagged to the secondary antibodies to the primary. And here's a sample of how the immunofluorescence appear. This is a test to show you. The highlighted is the immunofluorescence test. The next test is the flow cytometry. Flow cytometry is a strong and diverse analytical method for measuring the physical and chemical properties of cells or particles in a fluid suspension. It enables a single cell examination of several parameters, making it useful in immunological, hematological, cancer, and microbiological. In the application of immunophenotyping, surface or intracellular markers, the cell sorting use fluorescence activated cell sorting, or FASC, and the measurement of cell proliferation here using DNA binding dyes or markers that label newly synthesized DNA. So the apoptotic detection as annexin V binding or capsius activity, the cell wall integrity and the mitochondrial potential. So the measurements of cytokines is production and is used in the infectious high throughput and multi-parametric analysis, quantitative data, and single cell analysis. But the limitation of this flow cytometry may not be suitable for all samples because it's on top of it's expensive and requires skills, expertise skills. The next test is the radioimmunoassay. Is usually used as a radioactively tagged to a substance or to evaluate hormones, medication, and other antigen concentrations in a sample. It uses immunological and radioactivity to quantify tiny components that other approaches cannot be identified. So the applications of RIA or radioimmunoassay are to detect hormones, vitamins, serum, proteins, and infectious agents like hepatitis B surface antigens, antibodies, and medication. It detects a minute or as much as nanograms or picograms of that nature. This is the radioimmunoassay where we put a buffer into a tube, step one, and add known amount of unlabeled antigens to the mixture and to compete for the binding sites of the antibodies. Once the antibodies are competing to each other with the unlabeled amount of antigen, then we add a radioactive antigen to the mixture. Okay, and then we look at step 4. The added amount of the antibodies is fixed. Okay, the added fixed, and the radioactive step 5 with radioactive antigen is displaced from the antibody molecules by unlabeled antigen and precipitate antigen antibody complex with the PEG secondary antibodies. The step 6 will be the antibodies bound antigen are separated from the free agent in the supernatant fluids, and the radioactivity are measured from each one of them. The next method is the molecular techniques. In the molecular polymerase chain reaction of PCR, polymerase-based chain reaction, many molecular biological techniques uses PCR method to amplify DNA sequences. It is used for genetics, forensic science, medical diagnostics, and biological research, and including molecular biology and environmental sciences. It is used for detection of viral, bacterial, DNA, RNA, such as SARS virus, tuberculosis, human papilloma virus. And the PCR cycles are uses denaturation, annealing, and extension. Each cycle doubles the targeted DNA, causing an exponential amplification, usually 20 to 30 cycles, 40 cycles, producing millions or billions of DNA segments. Variations to PCR is the real-time PCR, reverse transcription PCR, multiplex PCR, and nested PCR. Next generation sequencing. This is very highly uh, uh, throughput applications to find the sequences of DNA or RNA. Applications of next generation sequencing is including quick sequencing of the whole genome, deeply seated targeted regions of performing RNA sequencing to identify the new RNA variation and splicing sites and quantify the mRNA for the gene expression studies. The assessment of epigenetic factors such as genome-wide DNA methylation and DNA protein interactions. This is the diagrammatic presentation of next generation sequencing workflow. So the step one will be the DNA extraction. The step two will be the DNA fragments in the library of preparation, and then the sequencing will be step three, and then ultimately it will be the analysis of the DNA extracts. In the next generation sequencing, the molecular biology studies the genome and the protein sequencing. The sequencing helps researchers to find the gene and the non-coding gene DNA alterations, diseases, and phenotypic connections, and the therapeutic targets, identification of the pathogens, including new and emerging infections, by sequencing the genome. The advantages of next generation sequencing is to screen more samples cost effectively and detect numerous variations in the specific genomic regions. But the downside of next generation sequencing is that common challenges in the next generation is basically the yield, the integrity, and the impurities. LAMP or loop mediated isothermal amplification. This is a test to amplify DNA with high specificity and efficacy at a constant temperature. Its application is to point of care testing for infections such as malaria and tuberculosis. CRISPR is based diagnostics stands for clustered regularly interspaced short palindromic repeats, a system to detect specific DNA or RNA sequence. The applications of this technology is really wide, and used in the fields of gene editing, functional genomics, diseases, research therapies for diseases like cystic fibrosis, cancer research, targeting specific mutants and therapeutic purposes, and drug development by creating and modifying disease models, infectious drug research by learning pathogens mechanisms, and generating new antiviral and antibacterial methods. And the safety concern or the downside of this CRISPR comes with several ethical and safety concerns like off-target effects like unintended editing may end up with harmful effects. So germline editing is a future and also raises ethical issues. This is the diagram that shows how the CRISPR or gene editing is done. If you can see, there's a part of the scissors to show that how the genes are edited. Next is the combination or combined techniques. In the combined testing, it involves serological and molecular assay. So this combination of serological basically is antibody-based and molecular and nucleic acid based. So this method improves sensitivity and specificity. Serological techniques involved enzyme-linked immunosorbent assay or ELISA test, Western blot, and immunofluorescence. In the molecular techniques involves polymerase chain reaction, real-time PCR, next-generation sequencing, and reverse transcription. So these combined investigative tests are applicable in infectious diseases like COVID, HIV, hepatitis, and for vaccine efficiency monitoring, genetic disorders, mutation detection, cancer-related genetic mutation, and identifying autoantibodies. So the advantage of this combined technique is suitable for infectious disease detection, cancer markers, genetic disorders, and autoimmune disorders for better sensitivity and specificity, and the disease staging and monitoring. Meanwhile, the downside of this are the regulatory and ethical issues, the cost, cross-reactivity, false positive test results, and handling. In this diagram, we have to show the molecular and serological testing using a throat swab and blood testing for from the same patient. So if you look at the upper side, we're testing the SARS-2 using the molecular techniques or mRNA, and the down one using the blood, using for IgG, IgM, and proteins, and we are using our serological method. In the combined other techniques will be the multiple assays. Multiple assay where it is simultaneously detecting SARS virus, influenza, respiratory virus, syncytial virus in a single PCR reaction to reduce time, increase efficacy to identify multiple pathologies or pathogens in the similar clinical presentation but with different treatment or public implications. Here's a sample of multiple assays. So we are targeting SARS, RSV, influenza A, influenza B, all simultaneously, so that we have a shortened period of time and multiple tests done at the same time. The advantages of multiple assays is the high throughput and efficacy, cost effectiveness, and comprehensive data collection, while the downside are the complexity of the assay design, like cross-reactivity, data interpretation, analysis with complicated data set, quality control, and lack of standardization. The next section of the combined method techniques will be the personalized medicine. In the personalized medicine, in the context of infectious diseases, involve tailoring prevention, diagnosis, and treatment strategies for individual patients based on their genetic makeup, the genetic characteristics of the pathogens, and other personal factors. Precision medicine in infectious diseases, the key aspects involve the pathogenic genomics, host genomics like COVID, pharmacogenomics, biomarkers of contagious diseases, and vaccine development. The application can be for rapid diagnosis, improved vaccine strategies, and targeted and personalized treatment. The challenges will be the downside will be the cost, access, ethical issues, regulatory and standardization issues. So this is a part of the personalized medicine where the omics-based disease signature, the biomarker values and differentiation, patient stratification and trial design, bedside decision support tools, exploratory clinical studies. So these will give us an understanding of how the infections can be in the social sector, health sector, and the WHO sector, where we can design personalized medicine for the clientele base. This diagram shows how personalized epidemiology can be conducted very specifically to the environmental factors involved with multiple people in the personalized medicine. Precision medicine involved in infectious diseases is the other sector we have looked into. So one of the sectors, the biomarker discovery. Biomarker discovery is using genomics, proteomics, metabolomics, immunological markers, bioinformatics, which enables accurate early diagnosis, disease progression, and therapeutic guidance. Some examples are we using C-reactive proteins, procalcitonin, HIV RNA levels to monitor the effectiveness of antiretroviral viral therapy, hepatitis B surface antigen for diagnosis monitoring. For SARS RNA for diagnosis and monitoring, the challenges and the downside is the validation, standardization, and the complexity of these tests. This diagram shows the biomarker discovery, the complexity of taking and finding out specific infections. Okay, for example, Borrelia. So from the monkey to the how the microbial antibody discovery, and then the particular Borrelia has been isolated in the protein assay, and then immunoprecipitant from either the species from samples from the urine or the serum has been done, and then we take it to the mass spectrometry, and then we list out the biomarker candidates very specifically through high potential biomarkers or potential biomarkers references. One more area is the artificial intelligence and machine learning. So there are several fields are engaged here, including genomics surveillance, pathogen evolution, disease detection, and diagnostic. medication discovery, vaccine development, predictive modeling, and outbreak surveillance. So the issue here will be the data availability and quality, as well as interpredictability among the difficulties with this model. That's the downside of this artificial intelligence and machine learning. This is a sample of the artificial intelligence machine learning where pathogens, and then we look at the transmission, then the medical experts are involved, the primary care, data collection, data science, artificial intelligence decision supporting, support, and the implementation subsequently. This is where the AI comes in. Proteomics and metabolomics. This is the area which is a very powerful omic technology currently widely used that gives an insight to proteins and metabolites in infectious diseases. So we get some insights into identification of pathogen proteins, biomarkers, host pathogen discovery, vaccine development, and metabolic profiling of the pathogens. This is a sample of where we are looking at the samples from the cells, enzymes, fluids, or the secretions, and then we go into extraction of the metabolites, and then subsequently we take that the statistics analysis, the pathway visualization through after it has gone through the spectrometry. What are the emerging and advanced techniques currently? One of them will be the digital PCR. There's an improvement in sensitivity quantification with partitions of samples into many individual PCR reactions. I'd like to show you an example of this. And here is a nasopharyngeal swab for an infection. And lysis, then an RNA has been extracted. The crude lysate has been separated. Then we can see that the plates there are individualized, and we can do multiple tests on these partitions. And the next one will be the mass spectrometry based proteomics. This is very widely used and used in infectious diseases like HIV or human immunodeficiency viruses, tuberculosis, bacterial infections like infections, Zika virus, malaria, COVID, influenza, hepatitis C, fungus infection, helminthic infection. The key functions are the pathogenic identification, bioidentification, immune response, and vaccine development. These techniques use shotgun proteomics, targeted proteomics, and isotope labeling. The challenges or downside will be the sensitivity, specificity, and data analysis and clinical interpretation. This is the diagram to show how mass spectrometry based proteomics works. So if you look at left upper part, you can see the sample of interest. It can be a fluid, or like a sputum or blood sample, or a secretion or urine has been isolated, and then it is mixed in a protein mixture, and then blended and excretion, and then it's put onto protein peptide mixture through the in-gel proteolytic digestive enzyme, and then it's taken out into mass spectrometry where it has been identified through a database. And this is how mass spectrometry based proteomics works. In conclusion, the future of immunogenic testing will be shaped by technology advancement, personalized medicine, artificial intelligence, and standardization, all of which are aimed to address the myriad of factors spanning from pre-analytical, analytical, post-analytical phases that influences the accuracy, reliability, and the immunological testing of results. These will enhance the effectiveness of immunological testing in the diagnosing and managing of infectious diseases and immune-related diseases. I am Dr. Radha Krishna, signing off. Thank you so much.